In the dimly lit laboratories of 1928 London, a humble Scottish bacteriologist named Alexander Fleming unwittingly changed the course of medicine. On a cluttered bench, amidst petri dishes teeming with bacterial cultures, Fleming noticed a peculiar phenomenon. A mold, Penicillium notatum, had begun to secrete a substance that killed the surrounding bacteria. He called it penicillin, a breakthrough that promised to revolutionize medicine. Yet, for all its potential, penicillin remained frustratingly elusive. Fleming's discovery, remarkable as it was, could not be translated into a practical treatment. Its yields were minuscule, unstable, and extraordinarily difficult to purify. For over a decade, penicillin remained a scientific curiosity rather than a weapon in humanity's fight against disease. Medical texts recognized its potential, but the practical limitations were enormous. Cultures grew slowly, the active compound degraded quickly, and extraction methods were crude. Laboratories in England attempted to coax larger yields from the stubborn mold, but success remained stubbornly out of reach. Fleming, Chain, and Florey, among others, would later be credited with the foundational science, yet none could imagine the scale on which penicillin would one day operate. The world, meanwhile, was barreling toward global conflict. As World War II erupted, the death tolls mounted not just from bullets and bombs but from infections that might otherwise have been minor. Wounds that could have been survivable were turning fatal due to gangrene, septicemia, and pneumonia. Infections claimed more lives than combat in earlier wars, a grim statistic that underscored the urgent need for medical innovation. Soldiers fought on muddy battlefields, often in conditions where even basic antiseptic measures could not prevent deadly infections. It was in this crucible of human suffering that penicillin would finally emerge from the shadows of the laboratory. The turning point came in the late 1930s and early 1940s, when a team at the University of Oxford, led by Howard Florey and Ernst Chain, managed to isolate, purify, and test penicillin in controlled experiments. Their approach was methodical. Chain's meticulous chemical analysis allowed the team to identify active fractions of the mold extract, while Florey's clinical insight guided the testing on both animals and, eventually, human patients. In January 1941, the team administered penicillin to a critically ill patient suffering from a life-threatening bacterial infection. The results were immediate and dramatic. Fever subsided, infection markers fell, and recovery began. This marked the first time penicillin had demonstrated unequivocal therapeutic value. Yet the Oxford team faced a formidable barrier. Producing enough penicillin to treat even a handful of patients was nearly impossible with their laboratory-scale techniques. Across the Atlantic, the United States had mobilized its industrial might for war, preparing not only tanks, planes, and ships but also medical technologies. In 1941, as the U.S. entered the war, government agencies recognized that penicillin could be a decisive factor in saving lives, particularly on the front lines. The challenge was daunting. The Oxford method produced mere milligrams of penicillin at a time. To supply entire armies, production needed to scale up by factors of thousands, or even millions. This required not just scientific ingenuity but industrial coordination at a scale unprecedented in medical history. Enter the pharmaceutical giants, Pfizer, Merck, Squibb, and Eli Lilly. These companies brought to the table expertise in fermentation, chemical engineering, and mass production, disciplines honed over decades for the manufacture of vitamins, insulin, and other biologics. Pfizer, in particular, pioneered deep tank fermentation, a revolutionary technique that allowed larger volumes of mold cultures to grow under controlled conditions. Unlike the shallow trays used at Oxford, these massive steel tanks, sometimes holding tens of thousands of liters, could sustain higher oxygen levels and precise temperature control, dramatically increasing penicillin yields. The scale was almost unimaginable. By late 1943, production had increased from mere grams to hundreds of millions of units per month. Coordinating this massive effort required the collaboration of government and industry in ways that were unprecedented. The U.S. War Production Board, alongside the Office of Scientific Research and Development, OSRD, oversaw contracts, allocated resources, and directed research priorities. Laboratories across the country shared samples, standardized testing protocols, and even swapped techniques for improving yields. A mold that produced modest amounts of penicillin in one lab could be enhanced by techniques developed elsewhere, a form of scientific teamwork that transcended individual companies and universities. Yet, the technical challenges were formidable. Penicillin was extraordinarily sensitive to environmental conditions, with slight changes in pH, temperature, or contamination rendering entire batches useless. Early production runs often failed, and the pressure to deliver effective doses to troops intensified as Allied forces planned the invasions of North Africa, Sicily, and eventually Normandy. Each failure was a race against time. A delay of weeks could mean thousands of preventable deaths. Scientists and engineers worked around the clock, developing purification techniques, scaling up fermenters, and testing formulations that could survive transport to distant battlefields. The stakes were deeply human. In the North African desert, medics treated soldiers with severe wounds, often amputating limbs to prevent gangrene from spreading. In field hospitals across Europe and the Pacific, infection remained the greatest silent killer. Every vial of penicillin produced represented a potential life saved. A soldier returned home, a wound prevented from becoming fatal. By mid-1944, the first large-scale shipments of penicillin reached Allied forces in Europe, marking a turning point in battlefield medicine. Suddenly, wounds that would have been fatal in previous conflicts could now be treated effectively. Mortality rates plummeted, and the psychological impact on soldiers and medics alike was profound. Penicillin was not just a medicine. It was a symbol of hope in a war defined by destruction. The mass production of penicillin was also a victory of industrial ingenuity. Deep tank fermentation, standardized testing, and interlaboratory collaboration became a blueprint for future pharmaceutical manufacturing. The rapid mobilization of resources, the integration of scientific discovery with industrial capacity, and the strategic application to wartime medicine demonstrated the potential of human innovation under pressure. It was a triumph not only for medicine but for the Allied war effort itself, a technology that saved more lives than many of the weapons on the battlefield. By the end of 1945, penicillin had firmly established itself as a transformative agent in both medicine and war. Millions of doses had been produced, distributed, and administered. Soldiers who would have died from septic infections survived to return home. The age-old specter of death from minor wounds had been, for the first time in history, effectively challenged. Fleming's mold, Chain's meticulous chemistry, Florey's clinical foresight, and the industrial power of American and British manufacturers together had rewritten the rules of survival in modern warfare. In the shadows of tanks, trenches, and hospitals, penicillin quietly, but irrevocably, changed the nature of war. Its story was not one of explosions and strategy, but of patience, precision, and perseverance. The quiet hero of World War II was neither a soldier nor a general, but a mold that had waited years for humanity to catch up, and the relentless humans who refused to let its promise go unfulfilled. As 1942 dawned over a world embroiled in total war, the promise of penicillin remained tantalizingly close yet maddeningly out of reach. Laboratory successes at Oxford had proven the drug's extraordinary potential, but translating grams of a fragile substance into doses sufficient to treat entire armies required industrial-scale ingenuity. The challenge was not merely scientific. It was logistical, bureaucratic, and monumental in ambition. The Allies needed a miracle, and penicillin was poised to deliver one. The U.S. government recognized that to win the war, science had to be mobilized like an army. In 1941, the Office of Scientific Research and Development, OSRD, led by Vannevar Bush, coordinated efforts to translate laboratory discoveries into practical tools for the battlefield. When penicillin's promise became undeniable, the OSRD orchestrated partnerships between universities, pharmaceutical companies, and government labs. The scale was unprecedented. Pfizer, Merck, Eli Lilly, and Squibb became industrial allies in a race against time, sharing techniques, strains, and insights in a spirit that blurred the line between competition and collaboration. The first major breakthrough came in understanding fermentation. At Oxford, penicillin had been cultivated in shallow trays, simple but inefficient. The method yielded mere milligrams, enough for small-scale experiments but wholly inadequate for the front lines. American scientists, particularly those at Pfizer, introduced deep tank fermentation, large aerated steel tanks where mold could grow under tightly controlled conditions. These tanks, sometimes holding tens of thousands of liters, were equipped with sophisticated oxygenation and temperature controls, designed to coax maximum output from the mold. The result was staggering. Yields multiplied by orders of magnitude, and production moved from laboratory curiosity to industrial reality. Yet deep tank fermentation was far from straightforward. Penicillin is notoriously sensitive. Slight deviations in pH, temperature, or contamination could ruin entire batches. Early industrial runs failed repeatedly. Engineers battled leaks, microbial contamination, and equipment limitations. To solve these problems, they borrowed techniques from brewing and chemical engineering, applying principles used in beer and vitamin production to this revolutionary drug. Each successful batch represented not just scientific achievement but a carefully orchestrated ballet of microbiology, chemistry, and mechanical engineering. Simultaneously, chemical purification became a critical focus. Raw fermentation broths contained a mix of mold, sugars, and bacterial byproducts that made penicillin unusable for medical purposes. Scientists developed extraction techniques using solvents and filtration, refining methods to maximize yield without degrading the delicate antibiotic. Even minor improvements, slightly more efficient filtration, better temperature control, or a more effective solvent, could translate into tens of thousands of additional doses for soldiers on distant battlefields. The race was not simply to make penicillin, it was to make it fast, in massive quantities, and reliably. Behind the machinery and chemical equations were people whose perseverance was as vital as any scientific advance. Chemists worked 12-hour shifts in laboratories coated with white tiles, pipetting, measuring, and monitoring cultures that could produce life-saving medicine, or nothing at all. Engineers inspected every fermenter, every valve, every gauge, ensuring the enormous steel vessels functioned flawlessly. And administrators coordinated the flow of raw materials, corn steep liquor, sugars, acids, from farms and factories across the country into pharmaceutical plants that hummed day and night. It was a war effort as complex and demanding as any battlefield campaign, and every failure risked lives in combat zones. By 1943, production began to scale in earnest. Pfizer's Brooklyn plant became the epicenter of deep tank fermentation, churning out penicillin in volumes previously unimaginable. Eli Lilly and Merck developed complementary methods, optimizing yields and standardizing quality control. Government oversight ensured that every batch met stringent standards, as contaminated or weak penicillin could be more dangerous than none at all. Within a few months, millions of units were being produced monthly, a monumental increase from the few grams available at Oxford just 2 years prior. The military impact was immediate and profound. In North Africa, during the Allied campaigns against Axis forces, medics began treating infections with a drug that could turn near-fatal wounds into survivable injuries. Soldiers with septicemia, gangrene, or pneumonia, once almost certain to die, found themselves stabilized, treated, and sent back to the front lines. Field hospitals that had once resembled triage for the doomed were transformed into centers of recovery and hope. The psychological effect on both soldiers and medics was immense. Penicillin was not just a medicine but a symbol of technological superiority and human ingenuity. Strategically, penicillin's mass production changed the calculus of war. Armies could sustain larger forces in the field because disease no longer claimed as many lives. Injured soldiers could recover and return to duty, preserving both manpower and morale. The Allies, particularly American and British forces, gained a subtle but decisive advantage. Where previously illness and infection had eroded effectiveness, now troops could endure, heal, and fight again. Penicillin became a silent force multiplier, amplifying the effectiveness of every soldier it touched. The story of scaling penicillin was also a triumph of global collaboration. Scientists in England continued to provide critical strains and data, while American industrial power turned those discoveries into mass-produced medicine. Government agencies, universities, and private industry coordinated like an enormous, complex machine, each part indispensable, each contribution magnified by collaboration. This model of cooperative innovation set a precedent for post-war pharmaceutical production, shaping modern drug development and laying the groundwork for antibiotics that would follow. By 1944, penicillin was no longer an experimental therapy, it was a standard of care on the front lines. Supplies flowed to Europe and the Pacific, reaching troops in Normandy during the D-Day invasions and across Pacific Islands during amphibious assaults. Mortality rates from infected wounds dropped dramatically. A soldier who would have faced almost certain death from gangrene in 1941 could now recover fully, often without amputation. Field medics could treat patients more aggressively, confident in the knowledge that penicillin provided a safety net. The transformation was remarkable not just for the lives saved but for the precedent it set. The war effort had demonstrated that large-scale pharmaceutical production was possible when necessity demanded it. It showcased the power of combining scientific discovery with industrial capability, a model that would later underpin the development of vaccines, antiviral drugs, and other medical breakthroughs. In effect, penicillin had inaugurated a new era, medicine as a strategic instrument of national power, capable of shaping not only health outcomes but also the trajectory of global conflicts. Yet even as production soared, the human dimension of penicillin's impact remained clear. Soldiers in makeshift hospitals, bandaged and feverish, received vials of a golden liquid that carried hope as much as healing. Nurses administered doses around the clock, monitoring patients for side effects while witnessing recoveries that seemed almost miraculous. Doctors marveled at the transformation of battlefield medicine. Wounds that had once meant inevitable suffering now healed, infections cleared, and lives were saved. The quiet heroism of those producing, transporting, and administering penicillin was every bit as vital as that of the men and women fighting on the front lines. By the end of 1944, penicillin was firmly entrenched as an indispensable tool of war. Its production had scaled from laboratory curiosity to industrial triumph. Its deployment had saved countless lives and enhanced military effectiveness in a conflict that spanned continents. And behind the statistics, the vials, and the steel fermenters, penicillin had delivered something more profound. Proof that science, industry, and human determination could converge to alter the course of history. By 1944, penicillin had transitioned from an experimental laboratory drug to a critical lifeline for Allied forces on multiple continents. Its journey from industrial vats to the battlefield was a story of logistics, ingenuity, and human perseverance, and it would profoundly alter the experience of soldiers and medics alike. For countless troops, the antibiotic became a quiet but decisive weapon in the fight for survival. The challenge of distribution was immense. Millions of doses were produced monthly, but getting them to front lines spanning Europe, North Africa, and the Pacific required an intricate network of transport, refrigeration, and coordination. Penicillin, unlike some more stable medications, was highly sensitive to heat and light. Improper handling could render it ineffective. Military logisticians developed specialized packaging and cold chain transport systems, ensuring the antibiotic could survive long journeys by rail, truck, ship, and even by plane drops to isolated units. Field hospitals were equipped with insulated storage, and medics were trained in proper administration techniques, transforming penicillin from a laboratory achievement into a practical battlefield tool. North Africa was among the first theaters to benefit from large-scale penicillin deployment. During the campaigns of 1942–43, Allied forces faced not only the Axis armies but also harsh desert conditions that exacerbated infections. Soldiers wounded in the arid, dusty terrain frequently suffered from infections that spread rapidly in wounds contaminated by sand and debris. Prior to penicillin, medics had little more than antiseptics and amputation to rely upon. Now, with the antibiotic in hand, infections could be halted before they became fatal. Reports from field hospitals described the miraculous recovery of soldiers who would have otherwise succumbed to septicemia. Mortality rates from infected wounds, which had once been devastating, dropped sharply. The Normandy landings of June 1944 marked another critical application. On D-Day and in the weeks that followed, Allied forces encountered heavy casualties from gunfire, shrapnel, and the chaos of amphibious warfare. Field hospitals, overwhelmed by wounded soldiers, administered penicillin as a standard component of trauma care. Surgeons observed that patients treated with penicillin experienced far fewer postoperative infections, even when surgeries were performed under less than ideal conditions. Medics noted a psychological boost among troops, who realized that a serious wound no longer carried the near-certain risk of death from infection. Penicillin had shifted the battlefield calculus. Survival depended less on luck and more on access to timely, effective medical intervention. The Pacific theater presented an even more daunting challenge. Soldiers and Marines fighting in jungles and on tropical islands faced a climate ripe for bacterial growth, where minor cuts and insect bites could escalate into life-threatening infections. Malaria, typhoid, and tetanus were already pressing concerns, and wound infections added a deadly layer of complexity. The delivery of penicillin across vast ocean distances required coordination between naval transport, field depots, and frontline units. Yet the effort was successful. Medics in the Philippines, Iwo Jima, and Okinawa administered penicillin to troops suffering from severe wounds, drastically reducing mortality rates and enabling soldiers to recover and return to duty. Individual accounts highlight penicillin's transformative impact. In letters and memoirs, soldiers frequently noted the relief of surviving injuries that would have been fatal in earlier wars. One medic recounted treating a soldier with a compound fracture contaminated by mud and debris. Within days of administering penicillin, the infection receded, and the patient's recovery was rapid. In countless hospitals, scenes that had once been grim, infected limbs, fevered patients, and desperation, were replaced with recoveries, hope, and renewed morale. Penicillin was not just a medicine. It was a psychological reassurance that survival was possible, even in the most hostile conditions. Field hospitals themselves evolved in response to penicillin's availability. Surgical techniques could be more aggressive, knowing that post-operative infections were far less likely to be fatal. Amputations, once a common response to gangrene, were now often avoidable. Medics and surgeons adjusted their practices, emphasizing wound cleaning and debridement while relying on penicillin to prevent infections. This shift not only saved lives but also preserved the quality of life for soldiers, reducing the long-term disabilities that had historically plagued veterans. The strategic implications extended beyond individual survival. By reducing mortality from infection, Penicillin allowed Allied forces to maintain higher troop strength and effectiveness over prolonged campaigns. Armies could sustain offensives with fewer replacements lost to disease, preserving both manpower and morale. The drug's impact was especially notable in campaigns involving rapid movement or extended supply lines, where traditional medical care was difficult to provide. Penicillin acted as a force multiplier, enhancing the operational capabilities of Allied units without a single shot being fired. In addition to its battlefield applications, penicillin had broader humanitarian significance. Military hospitals often treated civilians injured in bombings or affected by epidemics in liberated territories. The antibiotics' reach extended beyond the immediate goals of the war, saving lives and reducing suffering in occupied or war-torn regions. In doing so, penicillin demonstrated the dual power of science. It could serve military objectives while also preserving human life in broader society. The use of penicillin also prompted the emergence of a new medical culture within the military. Standard operating procedures incorporated antibiotic therapy as an integral part of trauma care. Training programs for medics emphasized the timely administration of penicillin, monitoring patients for adverse reactions, and coordinating with surgeons to maximize recovery. This professionalization of battlefield medicine reflected a broader understanding. The war was no longer fought solely with weapons and tactics, but with medical innovation, logistics, and rapid deployment of life-saving technologies. While penicillin dramatically reduced deaths from infection, it also highlighted the limits of contemporary medicine. Supply constraints meant that, particularly early in its deployment, the drug was reserved for the most severe cases. Less critical infections were treated with older antiseptics or sulfa drugs, emphasizing the necessity of judicious allocation. The tension between demand and supply spurred further innovation, driving improvements in production efficiency and storage methods. By the war's end, these advances had created a robust infrastructure capable of producing millions of doses monthly, setting a precedent for post-war pharmaceutical manufacturing. Across theaters of war, the narrative was consistent. Penicillin saved lives, enhanced operational effectiveness, and transformed the practice of military medicine. Soldiers who once faced a near-certain risk of fatal infection now had a lifeline. Field hospitals, once overwhelmed by preventable deaths, became centers of recovery. The drug's strategic and psychological effects were intertwined, reinforcing both the human and technological dimensions of warfare. Penicillin's impact on the front lines was profound, but it also foreshadowed a post-war revolution. Its success demonstrated that mass-produced medicine could alter the course of history. Governments and industries realized that scientific breakthroughs, when combined with industrial might and logistical planning, could have immediate, life-saving consequences. Penicillin thus became more than a medical milestone. It was a model for harnessing human ingenuity under pressure, a template that would influence pharmaceutical and military strategy for decades to come. In the crucible of battle, penicillin quietly reshaped the human experience of war. It did so not with the thunder of artillery, but with the silent precision of chemistry, microbiology, and industrial engineering. It saved lives, preserved armies, and provided hope where none had existed before. And in every field hospital, every jungle clearing, and every beachhead, the story of penicillin unfolded, not as a tale of machines or soldiers, but as a story of science meeting necessity at the moment humanity needed it most. Behind the massive steel tanks, vials, and field hospitals of World War II lay a network of individuals whose ingenuity, dedication, and perseverance made penicillin's mass production possible. While soldiers on the front lines received the immediate benefits of the antibiotic, the story of its triumph is equally a story of scientists, engineers, and administrators who transformed a fragile laboratory discovery into a life-saving industrial reality. At the forefront were the pioneering scientists who first recognized penicillin's potential. Howard Florey, Ernst Boris Chain, and Norman Heatley at the University of Oxford had meticulously isolated and purified the antibiotic, demonstrating its effectiveness in laboratory and early clinical trials. Their work required patience and precision. Each experiment demanded careful measurement, careful observation, and rigorous documentation. The team faced skepticism, limited resources, and the daunting task of producing enough penicillin for even a handful of patients. Yet their determination laid the essential groundwork for what would become a global industrial effort. Across the Atlantic, American chemists and engineers translated this foundational science into mass production. Pfizer, under the leadership of industrial chemists like Jasper Kane, pioneered the use of deep tank fermentation, a revolutionary method that allowed penicillin to be grown in tens of thousands of liters of nutrient-rich broth under precise conditions. Unlike the shallow trays of Oxford laboratories, these deep tanks allowed mold cultures to flourish on an unprecedented scale. Oxygenation, temperature control, and sterility had to be maintained with near-perfect precision. Even minor contamination could destroy entire batches, underscoring the delicate balance between human skill and technological innovation. Merck, Eli Lilly, and Squibb contributed complementary methods and innovations. Each company developed specialized purification processes, standardized quality control measures, and optimized fermentation techniques. Government agencies, particularly the Office of Scientific Research and Development, OSRD, coordinated these efforts, ensuring that knowledge, strains, and best practices were shared efficiently. This unprecedented collaboration blurred the lines between competition and cooperation, creating an industrial ecosystem focused singularly on producing life-saving medicine at massive scale. The human element extended beyond laboratories and factories. Production workers, often unseen in the grand narratives of war, worked tirelessly in challenging conditions. They monitored tanks, measured pH levels, cleaned equipment, and ensured sterility in environments where a single error could mean the loss of hundreds of thousands of units. Shift work, long hours, and the constant pressure to meet military demands made the production of penicillin a human as well as technological triumph. Each vial of penicillin was a testament to both the precision of machinery and the dedication of the people operating it. In parallel, the military integrated penicillin into medical practice with unprecedented efficiency. Surgeons, nurses, and medics were trained in its proper administration, dosing, and monitoring. The antibiotic transformed battlefield medicine. Wounds that previously demanded amputation could now heal. Infections that once led inevitably to death were now preventable. Medics adjusted surgical techniques, confident that post-operative infections could be managed. Hospitals shifted from reactive centers of triage to proactive hubs of recovery. The human ingenuity in administering penicillin complemented the technological breakthroughs in its production, creating a seamless chain from laboratory to battlefield. Technological innovation went hand in hand with human skill. Deep tank fermentation required the precise measurement of oxygen levels, nutrients, and temperature. Purification involved careful extraction using solvents that could easily destroy the delicate antibiotic if mishandled. Even transportation posed challenges. Penicillin needed to be protected from heat, light, and contamination as it traveled from factories to distant fronts in Europe, North Africa, and the Pacific. Engineers designed refrigerated containers, insulated packaging, and logistical networks capable of delivering medicine under combat conditions. The result was a feat of coordination that rivaled the complexity of any military operation. Beyond technical and logistical achievements, the success of penicillin represented a cultural shift in science and medicine. Collaboration became a new norm. Universities, corporations, and government agencies shared knowledge freely, prioritizing human lives over proprietary advantage. Laboratories across continents exchanged strains, methods, and data, creating a global network of innovation. This spirit of collaboration not only accelerated penicillin production during the war, but also set the stage for post-war pharmaceutical research, demonstrating that cooperation between science and industry could achieve results on a planetary scale. The psychological impact on soldiers was equally profound. For the first time in military history, wounded troops could recover reliably from infections. The threat of death from a minor scratch or deep laceration diminished. Morale improved, and confidence in medical care strengthened. Soldiers witnessed firsthand that human ingenuity, embodied in the quiet work of scientists and factory workers far from the front lines, could directly influence survival. Penicillin became a symbol of hope, a tangible proof that technology and human effort could bend the odds in life-or-death situations. The industrialization of penicillin also had long-term implications for medicine. Techniques developed during the war, such as deep tank fermentation, sterile processing, and large-scale purification, became the foundation for modern antibiotic production. The war accelerated the adoption of industrial-scale pharmaceutical manufacturing, demonstrating that scientific discoveries could be rapidly translated into accessible, life-saving medicines. The successes and failures of the wartime penicillin program informed practices for future antibiotics, vaccines, and biologics, shaping the trajectory of modern medicine. Yet the human story remained at the heart of the triumph. Scientists labored for months refining processes. Production workers endured exhausting shifts ensuring consistency and quality. Medics and surgeons applied the medicine under extreme conditions. Every milestone represented countless acts of precision, patience, and courage. The synergy between human effort and technological advancement transformed penicillin from a fragile laboratory curiosity into a strategic asset that saved countless lives and changed the nature of warfare. By 1945, the combined efforts of scientists, engineers, factory workers, and military personnel had achieved what once seemed impossible. Millions of doses of penicillin were available to Allied forces, turning the tide against infections that had long been as deadly as bullets. The war had demonstrated not only the power of scientific discovery, but also the capacity of human determination, collaboration, and innovation to transform lives on a massive scale. Penicillin's journey, from mold in a petri dish to vials in the hands of medics, was more than a technological achievement. It was a testament to human ingenuity, perseverance, and the ability to harness science for the greater good. The drug saved lives, preserved armies, and reshaped medical practice. It proved that in the crucible of war, innovation and human determination could converge to produce outcomes that were nothing short of miraculous. In the end, the human and technological triumph of penicillin during World War II was inseparable. The delicate mold that Alexander Fleming discovered in 1928 found its destiny not solely through chemistry, but through the relentless dedication of countless individuals who believed in the promise of science and the value of life. Together, they transformed a laboratory breakthrough into a force that reshaped medicine, saved millions of soldiers, and redefined the limits of what could be achieved when human ingenuity met necessity. As World War II drew to a close in 1945, the industrial, scientific, and human efforts that had scaled penicillin to unprecedented levels began to ripple far beyond the battlefield. What had begun as a fragile mold in a London laboratory had become a symbol of human ingenuity, a practical weapon in the fight against infection, and a model for modern medicine. The legacy of penicillin was not merely in the lives it saved during the war, but in the transformation it brought to healthcare, pharmaceutical production, and public health worldwide. The immediate post-war impact was profound. Soldiers returning from Europe and the Pacific carried with them stories of survival made possible by penicillin. Amputations that might once have been necessary were avoided. Infections that would have claimed lives were now treatable. Field hospitals, once places of desperation, had become centers of recovery, demonstrating the dramatic shift in medical outcomes. Civilian hospitals, too, quickly adopted penicillin, recognizing its potential to treat everything from pneumonia and strep throat to more severe bacterial infections. The antibiotic revolution had begun. Mass production techniques developed during the war continued to evolve, laying the groundwork for the modern pharmaceutical industry. Deep tank fermentation, refined purification methods, and rigorous quality control practices became standard in post-war antibiotic manufacturing. Pharmaceutical companies applied the lessons learned from penicillin to other emerging drugs, accelerating the development of streptomycin, tetracycline, and later generations of antibiotics. The rapid translation of laboratory discoveries into scalable, effective therapies became a blueprint for modern drug development. Penicillin's success also reshaped public health priorities. Governments and medical institutions recognized the potential of antibiotics to dramatically reduce mortality and morbidity from infectious diseases. Vaccination programs, hospital protocols, and sanitation standards were strengthened, creating a more resilient public health infrastructure. The war had shown that investment in medical science could yield tangible benefits in both military and civilian contexts, a lesson that would drive post-war health policy for decades. The societal impact was equally significant. For the first time in human history, a large-scale, mass-produced drug offered near-universal protection against a broad range of bacterial infections. Communities previously devastated by epidemics now had access to a reliable treatment. Families could survive illnesses that had once been fatal, and public confidence in modern medicine soared. Penicillin became more than a drug. It became a cultural symbol of hope, progress, and human ingenuity. In the scientific community, the wartime penicillin effort had a lasting influence on research culture. Collaboration across universities, government laboratories, and private companies demonstrated the power of cooperative innovation. The rapid sharing of techniques, strains, and data during the war created a precedent for future research efforts, fostering an environment in which scientific discovery could be accelerated through shared knowledge. The ethos established during the penicillin project would later influence large-scale scientific collaborations in fields as diverse as molecular biology, virology, and vaccine development. Penicillin also reshaped medical education and clinical practice. Physicians trained during and after the war incorporated antibiotics as a fundamental part of treatment. Surgical techniques evolved with the knowledge that postoperative infections could be effectively managed. Medical curricula emphasized microbiology, pharmacology, and the integration of laboratory science with clinical care. The success of penicillin provided a concrete example of how rigorous scientific research could directly inform and improve patient outcomes, bridging the gap between theory and practice. The legacy of penicillin extended into global health as well. International relief efforts, colonial health programs, and post-war reconstruction initiatives incorporated antibiotics into their strategies to combat infectious disease. Diseases that had plagued populations for centuries, pneumonia, syphilis, strep infections, could now be treated reliably. In many countries, mortality rates from bacterial infections dropped sharply, marking a new era in public health. The foundations laid during wartime production enabled rapid response to epidemics and created the infrastructure necessary for future pharmaceutical distribution on a global scale. Beyond medicine and public health, penicillin's story became a symbol of what humanity could achieve under pressure. The collaboration of scientists, engineers, government officials, and industrial workers during the war demonstrated the power of coordinated effort and the translation of abstract discovery into tangible impact. It showed that necessity could drive innovation, that technology could amplify human capability, and that perseverance and ingenuity could overcome seemingly insurmountable challenges. Penicillin became a case study in the intersection of science, industry, and society, a model for future technological revolutions. The wartime experience also prompted reflection on ethical and social responsibilities in medicine. The rapid deployment of penicillin raised questions about access, equity, and the role of government in ensuring that life-saving treatments reached those in need. Lessons learned during World War II informed later debates about the distribution of medical resources, the regulation of pharmaceuticals, and the balance between profit and public good. Penicillin's success underscored the importance of viewing medicine not only as a scientific endeavor but as a societal responsibility. By the late 1940s, penicillin had firmly established itself as a cornerstone of modern medicine. Its development and mass production had transformed the treatment of infections, reshaped surgical practices, and saved millions of lives. It had altered the experience of war, providing soldiers with protection against the once certain threat of fatal infections. It had strengthened public health infrastructure, guided pharmaceutical innovation, and inspired generations of scientists, engineers, and medical professionals. In retrospect, the story of penicillin during World War II is not simply a tale of science or technology. It is a testament to human determination and ingenuity. From Fleming's accidental discovery in 1928 to Florey and Chain's laboratory breakthroughs, to the industrial might of American pharmaceutical companies, to the medics administering doses on distant battlefields, penicillin's journey embodies the intersection of knowledge, skill, and perseverance. It demonstrates that the impact of science is magnified when coupled with collaboration, resourcefulness, and the will to meet urgent human needs. The legacy of penicillin endures to this day. Antibiotics remain a foundational element of medicine, and the lessons of wartime production continue to inform modern pharmaceutical practices. The success of the penicillin project reshaped expectations of what medicine could achieve, establishing a new standard for speed, scale, and impact. And while the war that necessitated its mass production ended decades ago, the human and technological triumph it represented continues to save lives and inspire innovation worldwide. In the end, penicillin stands as a reminder that even in humanity's darkest hours, discovery, collaboration, and determination can illuminate a path forward. From mold to medicine, from laboratory to battlefield, from industrial vats to global health systems, penicillin's journey transformed the modern world. It is a story of survival, ingenuity, and hope, a legacy that transcends war and endures as a testament to what human effort, when combined with scientific brilliance, can achieve.